Good morning, children. In this session, we will take up the fifth experiment to demonstrate anaerobic respiration. Let's understand the setup of this experiment. We will have an experimental setup and a control setup. In the experimental setup, we are going to take a test tube which is filled with mercury and insert soaked and peeled off peas into the test tube filled with mercury. And then we are going to invert this test tube in a beaker of mercury. Here the seeds will float at the top and will be completely surrounded by mercury. Now children, I had mentioned peeled off peas. Now why do we need peeled off peas? The peas, when they respire, they will produce carbon dioxide, and this carbon dioxide needs to be diffused out. So for faster diffusion process, the peas are peeled off. Now let's see what is the control setup. In the control setup, we will take a test tube which is filled with mercury, and in this test tube we are going to push some boiled peas, that is, they are killed by heating, and we are going to sterilize these peas with some antiseptic. As you had earlier studied, we need to sterilize these boiled seeds with some antiseptic to prevent bacterial growth. Now, the similar way, we are going to invert this test tube in a beaker of mercury. So this is the control setup, and then we are going to leave this setup for 2 days. What are we going to observe after 2 days? We will observe that the level of the mercury in the test tube of the experimental setup will fall, whereas the level of the mercury in the control setup does not fall. Now we need to know why there is fall in the level of the mercury. The peas have respired. After the respiration process, carbon dioxide is liberated, and this carbon dioxide has occupied the space in the test tube. Whereas in the control setup, we had pushed boiled seeds. They were dead seeds. The dead seeds have not respired, so the mercury level shows no change. Now, how do we know that the space occupied in the test tube is carbon dioxide gas? To test for carbon dioxide, we need to introduce a stick of potassium hydroxide into the test tube. This will float up through the mercury and once coming in contact with the gas will absorb the carbon dioxide, and the level of the mercury will again rise. This experiment shows that the peas have respired anaerobically, that is, without the presence of oxygen. Let us proceed and understand the difference between photosynthesis and respiration. Photosynthesis process, it occurs in the presence of chlorophyll. That means the cells which contain chlorophyll photosynthesize, whereas respiration process occurs in all living cells. Photosynthesis can take place only in the presence of light, whereas respiration takes place all the time, day and night. We use carbon dioxide and water as reactants for photosynthesis, whereas in respiration, glucose and oxygen is used. And the products of photosynthesis is oxygen, whereas in respiration the product is carbon dioxide, just the opposite of photosynthesis. In photosynthesis, light energy is converted into chemical energy and stored, whereas in respiration, chemical energy is partly converted into heat and partly into useful energy for helping us to perform various activities. Photosynthesis results in gain in weight because food is produced, whereas respiration results in weight loss because food is broken down. In photosynthesis, food is manufactured, that is, it is a constructive process. This process is called anabolic. Whereas in respiration, food is broken down, oxidized. This is destructive process and it is called catabolic. With this, we complete the chapter Respiration in Plants. Children, please answer the objective questions given at the end of the chapter for better understanding. Take care. Thank you.